Okay, welcome to Chapter 13, Modern Understandings of Inheritance. Today we're going to talk about some genetic ideologies that happened after Mendel and how this affects us as humans. So first let's talk about the chromosomal theory of inheritance. A lot of different scientists and geneticists contributed to this theory, Bovary and Walter in particular here pictured. The chromosomal theory of inheritance identified chromosomes as genetic material that were responsible for Mendelian inheritance. And this was actually proposed before direct evidence was made that chromosomes carry traits. So it's interesting that they were able to pinpoint that chromosomes as a theory were responsible for Mendelian inheritance, but they had never seen chromosomes carrying traits. So this was consistent with Mendel's laws. Thomas Hunt Morgan also contributed to this theory with his work with fruit flies, or Drosophila melanogaster. It was also found that alleles positioned on the same chromosome are not always inherited together. Linked genes can become unlinked during meiosis, which is a little bit different than Mendel had originally stated. We have previously talked about homologous recombination or crossing over. In 1909, Franz Janssen observed chiasmata, which is the point where chromosomes are in contact with each other and where they may exchange segments. Alleles can become unlinked and chromosomes can exchange segments, and this is homologous recombination or crossing over. Parental types are going to exhibit the same allelic combinations as parents, and non-parental are going to be the result of crossing over. There will be a difference in alleles than the parent type because of this crossing over effect. However, there was no technology to demonstrate crossing over. So an undergraduate student, Alfred Sturtevant, in 1913, was able to mathematically prove linkage and recombination. He created the first chromosome map, which is a linear representation of gene order on a chromosome. So he was able to infer gene order, and he discovered that alleles that are farther apart are more likely to dissociate in meiosis. And on the other side, alleles that are closer together are more likely to be carried together when crossing over during meiosis. Recombination frequency is the average number of crossovers between two alleles. Every type of allele combination is represented at equal frequency. In a 1931, Barbara McClintock and Harriet Creighton demonstrated crossover of homologous chromosomes in corn plants. So think back to week 1 when we were talking about women scientists that were Nobel laureates. Some of you picked Barbara McClintock, and now it's full circle. Karyotypes are the number and appearance of chromosomes and includes the length, banding pattern, and centromere position. Karyograms are visual representations of individuals' chromosomes. So usually when geneticists are looking for genetic mutations in the offspring of humans, they will look at karyograms to identify any chromosomal aberrations. Here you can see both autosomes and sex chromosomes, autosomes being any of the somatic or body chromosomes. Sex chromosomes determine sex, X and Y. Nondisjunction is when homologous chromosome pairs or sister chromatids fail to separate during meiosis, which can cause things like duplications or deletions and ultimately changes in phenotype. So this can occur during meiosis 1 or meiosis 2. Aneuploidy, so this is an aneuploid. Aneuploidy is an error in chromosome number. So if you're euploid, then you are an individual with an appropriate number of chromosomes for your species. If you're aneuploid, then there's an error in chromosome number. In monosomy, there's a loss of one chromosome, or trisomy, there's a gain of an extra chromosome. Down syndrome is often termed trisomy 21 because it's the 21st chromosome that has a multiple. There's an increased incidence of Down syndrome with an increase in maternal age when giving birth. Anybody over the age of 35 or older is considered to be a geriatric pregnancy, so with them, they tend to have a higher incidence of babies born with Down syndrome. Polyploidy is when there's more than the correct number of chromosome sets. This is, in animals, these animals are typically sterile, but in plants this is very common and actually makes them larger and more robust. X inactivation occurs when information is condensed into a quiescent structure or bar body, and this can happen randomly. This can be observed in torticats, which is shown by color variegation. In humans, there's a couple of examples of this. Triple X syndrome, where there are 3 X chromosomes. This individual would be phenotypically female, and there would be developmental delays and likely a lack of fertility. In Klinefelter syndrome, this is when an individual has XXY chromosomes. This individual is phenotypically male. They tend to have small testes, enlarged breasts, and a lack of body hair. And they can even have as many as 5 X chromosomes, and some variations of Klinefelter. Turner syndrome is when there's just 1 X chromosome, there's nothing else. 0 is usually shown, or O to hold the place of what should be another sex chromosome. So again, this individual is phenotypically female. They would have a short stature, a webbed neck, hearing and cardiovascular impairments, and typically they are sterile. Duplications and deletions can occur when chromosomal segments duplicate or delete themselves. This Cry-du-Chat syndrome. So this literally translates to cry of the cat, and it's because infants who have this typically have a very high-pitched cry, and they tend to have some nervous system abnormalities as well. This is caused by a deletion of 5p on a chromosome. You can also have chromosomal inversions or translocations. Inversions will be a 180-degree rotation of a chromosomal segment. This will likely have mild effects on the individual who has this. Inversions can be pericentric, where they include the centromere, or paracentric, where they exclude the centromere. Translocations is an entirely different situation. It's where part of a segment of a chromosome is taken from one part and transferred to an entirely other part. This can either be benign or devastating. It's implicated in a lot of cancers and even schizophrenia.